All right, so we'll get going. All right, good afternoon, everyone. Welcome to the Biology Department seminar of the Spring 2021 virtual edition of our Biology Department seminar series. So today I'm really pleased to welcome today's speaker, Dr. Mike Sears, to UMass Boston, even if only virtually. We wish it were in person, but obviously that's not what we're doing right now. Dr. Sears is joining us today from Clemson University in South Carolina, where he's associate professor in the Biological Sciences Department. Dr. Sears' research is focused on the thermal ecology of animals, particularly reptiles and amphibians. And he's especially interested in how animal species respond to environmental change, integrating a lot of different sort of layers of research onto this question, ranging from, I know, physiology, modeling, remote sensing data, genetic data, and so on. Dr. Sears has been really successful in his research career. He has dozens of high-impact publications in top journals, such as Ecology Letters, PNAS, Proceedings B, the American Naturalist, and others. I'm really pleased that Mike has agreed to give a seminar in our series today. Obviously, you know, we would rather it be in person, but since we can't do that, I'd like to just give Dr. Sears our warmest possible virtual welcome, which is a little bit hard to do, but we could use the clap emoji and so on. And thank him for taking the time to speak to us today. The last thing that I'm going to mention before he gets underway is that we have a social hour after the seminar, and where we can continue sort of in a more informal way any discussion about Dr. Sears' talk. So I'll post information about how to join the meeting in the chat. And we will do the formal question and answer after the seminar through the chat. So if you have questions, I think the protocol is to just post them into the chat. Is that correct, Brooke?
No. So if you have questions, please use the Q&A function. You can ask them anytime during the seminar. We'll just use them at the very end. So the Q&A function, you can find that on the bottom of your Zoom window if you haven't used it before.
Yes, sorry. I stand corrected. Use the Q&A for I'll post the happy hour list. It's happy hour link in the chat, and questions should go into the Q&A. And then also we can have, if you prefer to reserve your questions for the happy hour, you're welcome to come to that, and we can ask them, because that'll be a regular Zoom meeting rather than a webinar. And so you can ask your questions directly to Mike at that time in the happy hour as well. And so without any further ado, I'm going to pass over the podium to Dr. Mike Sears, who's going to talk about forecasting winners and losers of climate change, insights from the interactions between organisms and their microclimates.
Okay, thanks, Liam. Yeah, I wish I could be up in Boston right now. I miss the snow. We don't really get much of that down here in South Carolina, but maybe it's more of a pain than it seems. All right, so I guess today I'm going to give an overview of some of the work I do. You can see here on this graphic, this is sort of represents an overview of some of the work I do, where we take patterns of global climate, we downscale it to local microclimates, and we try to do this in a spatially explicit context to see how animals respond behaviorally and physiologically, and then from those responses predict sources and sinks across some larger landscape and try to predict things like species range dynamics. For that work, we really focus right now on about 4 different systems. One, we look at how the thermal landscape affects animals' ability to thermoregulate, how that affects individual physiologies. But then we also look at how interactions amongst individuals are mediated by that spatial heterogeneity. And we're beginning also to look at species interactions, such as predator-prey dynamics in that. But aside from adults, we also look at the consequences of the thermal environment on developing embryos. And then in other systems like plethodon salamanders, we're looking at how physiological plasticity may affect animals' ability to respond to climate change. Today, I'm going to focus mostly on adult thermoregulation and some embryonic physiology to try to tie it together a bigger story about how we're trying to push the limits of our species range models these days. So just quickly, and I'm going to have quite a few slides today, so I'm going to try not to dwell too much on any one. But quickly, the big problem in the world today is climate change. If you look over on this panel, you'll see that plotted are historic temperatures. We can just take North America on land. What this blue shaded area would represent would be the predicted temperatures given that humans aren't having any inputs with things such as greenhouse gases. And then this pink one would be the prediction you would make for temperatures if those greenhouse gases were included. And then you'll see this line, that's the real data. And no matter where you look at on the planet, you'll notice that that line of real data seems to go through the line that you would predict with human inputs. Now, what that suggests then is that temperatures are going to increase sometime into the future. And depending on whether we can keep the status quo or things get worse, what you would predict is there's going to be a higher risk of extinction for species across the globe. So it's sort of imperative for ecologists then to be able to predict which species are more at risk and what that might mean for ecological systems. Now, when you're trying to put together a predictive model, I tend to favor mechanistic models where we can put in actual real biology. And Mark Urban did a really nice job in Science a few years ago reviewing all the different things that should probably go into a mechanistic model. We should, of course, look at how environments are going to change. Lots of us have focused a bit on physiological responses. We probably should know something about the age-specific survivorships and reproductive rates of organisms, their ability to disperse and how they interact with others, but then hopefully also look at evolutionary responses. Now, Mark also did a literature review and noted that for quite a number of models that he looked at, most of them contained no mechanism at all. So these might be things like maxent models that some of you may be familiar with. But then, some smaller percentage, maybe 20%, only had one or more mechanism. And, that means everyone else is just focusing on one. So you'll notice that really, in terms of mechanistic understandings of future dynamics, we don't really know much. Now, my first sort of deep dive into this type of work was through an NCEAS working group headed by Lauren Buckley. And we had a whole room of people, I guess maybe 20 people, and we tried to come up with an organism or two that we could actually compare some of these correlative model approaches versus these mechanistic approaches. And we only came up with two species. One was a skipper butterfly, and the other was the best research organism on the planet, eastern fence lizards. Despite their drab appearance, they've been quite influential in our understanding of thermal ecology. But what we did is we tried to predict the range of this species. So here, this line is sort of a accepted range from field guides and things. And what we tried to do is with different mechanistic approaches, whether we looked at biophysical thresholds for some life stage, or we included correlations with life history, or these models that use activity time to predict foraging dynamics. What we found was that really, depending on the assumptions of the model, sometimes we got okay predictions, but most of the times we got really not great ones. So the gray areas are where the model predicts things should be. And then we actually made a prediction for the future where we add 3 degrees to the model as air temperatures. Now, what you find is that you always see a northern expansion of the range with climate change. So that was good. And that's something that the correlative models couldn't do. But the correlative models were really good at finding the current range, but not the future. So as someone who studies mechanism in biology, I was really frustrated that we couldn't figure out, we couldn't get a consistently good prediction with these models. And so much of the research that I've done since that group is really trying to take some of the assumptions that go into these models and relax them and see, just how much information do we need to know to make better prediction. And so why do our models fail? There are sort of 3 general reasons, and some of them are more severe than others. So one is environmental data are often poorly resolved. Historically, we were looking at cells in a GIS context of maybe, if you were lucky, 30 square kilometers on a pixel. that's coming down now, and with the use of drones, maybe this is becoming even less of a problem. But the real problematic data are things like the physiological data. And you'd think that over the last 30 years that we've been looking at the thermal sensitivity of pretty much any physiological response to temperature, that we'd have good data, except for we measure most of those things at constant temperatures. And recently we found that when you measure those same traits over variable temperatures, you can't really predict the right response. You can be off by 50% on something like a developmental rate or a growth rate. So not too helpful. So we have to really think about how we measure physiology in an ecological context. And then the other thing is that actual interaction between organisms and environment are usually ignored. We just assume that environments are homogeneous, and we'll pull data, whether it's temperature or humidity, ask how the physiology of an animal would respond, and we go with it. But animals also have behavior, and we have to think about that in the spatial contexts as well. All right, so quickly, I'm sure not everyone here has played around with species range models, so I'll give you just a quick primer. And basically, you'll pull down climate from some data set, and then you'll transform that climatic data into something meaningful for your organism. With lizards, we calculate something called operative temperature, which dictates what an animal's body temperature could be. And from that, we can estimate when an animal could be active at the surface, during which time it's eating. From that, we can calculate how much intake it has. We can get rid of the expenditures. If there's energy, if it can meet its energetic demands, it survives. Okay, you can live there. If the animal has excess energy, it might even be able to reproduce. And from that, we can estimate fitness. And we basically do this for every possible spot, you know, on a map. And that tells us where the species might be. Very basic. Now, we're going to focus first off today on this issue of activity, because one of the assumptions that you get is, let's just say you're working with kilometer scale data. You'll assume everything within that square kilometer is the same in terms of temperature, but we all know as field biologists anyway that it's a bit more variable on the ground. So I was really interested in knowing, like, what does that variability do to model predictions, and you know, how far off can these things be? So we went down sort of this long rabbit hole. But first, I'll show you, you know, how do animals regulate? So basically, if you look across the times of day, you know, the temperature's warm, they cool. An animal's going to have some minimum body temperature that it's willing to be active over. It's going to have some maximum temperature where it's going to be active. So, you know, it has some range. Let's see here. There we go. Now, the environment, for an environment, this would be a cool environment. There's going to be some minimum temperature in the environment the animal can exploit. This would be, say, in full shade. There's going to be a maximum temperature in full sun. And then where these lines sort of intersect is going to determine where an animal could be active. So as that warmest temperature approaches the minimum temperature an animal would be active over, it's active, and it can remain active as long as there's some overlap. And then once that maximum temperature cools down to a level the animal doesn't want to be active, that determines its rate. Now, if we look at a little bit warmer environment, you'll notice that the activity gets a bit longer, but they can still be active over the whole time of day. But if we go to an even warmer environment, now you might lose activity. So you have maybe a bimodal period, so a morning period, an evening period, but there's going to be some midday period where animals couldn't be active at the surface because they would overheat. And so you get some nonlinear dynamics in some sense. Now, that's a real simple sort of solution, assuming that animals have equal access to temperature in their environment. But real environments aren't that simple. So here's a sort of a heuristic view about how space might influence an animal's activity. So here is some habitat with approximately 25% of the environment that has temperatures that are favorable for animal activity. Now out here, so this is, you know, a nice shady bush, but it has the right temperatures. Out here, this is inhospitable sand. If you spend too much time out there as a small lizard, you'd overheat. So effectively, if you live in a habitat that looks like this, you can only be active in about a quarter of the habitat. But if we split that up, it could even get worse for you. So it could be that, you know, this space is too far to travel before you would overheat. So now you only have 1/16 of the habitat available for activity. Now in this configuration, this is just far enough that you can cross. Now you can access the whole bit of habitat, and then nature is messy. Now, the problem with this is, even from an empirical perspective, when people go out and put data loggers for temperature, you'll put, you know, randomly put things across the environment. All four of these habitats would look the same to you in terms of data, But in terms of the reality on the ground and how animals are interacting with these patches, it would be quite different. So that got me thinking about, well, what would those impacts be? And so I started diving into some models of animal movement, but giving them some choice of what patches to move amongst by thermoregulatory decisions. So I can basically take an animal, plop it down in some environment and say, go out and thermoregulate, move to do so. We have some rules, and those rules aren't necessarily important, but basically they're just trying to keep their body temperature within a certain range without overheating. And I can keep track of their body temperatures, how far they move, and how much energy it takes for them to do that. Now, the nice thing about computers is I can actually create very specific distributions of temperature. So I can actually create that little green area that has temperatures that overlap the animal's preferred temperature, and I can put them on very specific parts of that plot. I can increase the amount of good habitat, and I can also divide up the habitat into different patches, right? So 1, 4, and 16. I can also take these other temperatures and I can heat them up. I can cool them down and do every combination of cool and warm environments. And so I can put this animal in, put any number of these virtual lizards into their natural environments here and ask how well they thermoregulate. And when you do that, you find some really interesting results. And you find that things like availability of habitat, heterogeneity, and the dispersion of the habitat really affect thermoregulation. So here are two types of plots we'll look at. Here is this black line is the strategy for an animal that's not thermoregulating. So I just released it into the virtual arena and allowed it to move randomly and with the same rules that the other animals had, but they just weren't choosing it based on temperature. This is the temperature outside the hospitable habitat. So I was warming up those areas outside the green. This axis is measuring what I call thermoregulatory inaccuracy, or basically how many degrees above the animal's preferred body temperature did that animal get. So what you'll notice first, when we have just a small amount of habitat that's in the preferred zone, a conforming animal, basically its body temperature increases linearly with the environment, a 1-to-1 correspondence. But what's interesting is if you have one big patch, basically it's about the same as a conforming organism, even though it's trying to regulate, because it gets off that patch, it could get lost out in the bad habitat for longer periods of time. But then the more patches you add, you start getting a little bit better thermoregulatory performance. So these animals are only about 5 degrees off where they want to be in the most extreme environment, where these are 10 degrees off. And down here, you can see a corresponding plot showing the energy expenditure. So this would be the energy expenditure just to be a conformer. And then by trying to regulate, you can see these other ones. And what you find is when you have more patches, because there's a smaller distance between patches, they expend less energy for movement. Now, if we go to this other extreme and we add, say, 40% of the habitat is shaded, now what you'll notice, at least for these things, is quite interesting. The one patch habitat still isn't great, but now for the 4 and 16 patches, they can thermoregulate as well as they could under any circumstance. So they're basically perfect regulators across a broad range of temperatures. And so that heterogeneity really means something to them. And if you look at the energy expenditure to do that, again, it's reduced because there's a smaller amount of distance between patches. And when you look at the thermal variability within patches, you see a similar type of thing. If you have low variability, so no matter where you go, it's roughly the same temperature within a patch. The one patch environment looks like a conforming type of environment, whereas the 4 and 16 give you a little bit better thermoregulatory ability. But if you make a high variability of temperatures within a patch, all of a sudden the animals can do quite well and the energetic expense is low again. Now, I did these simulations for animals from 1 gram to a kilogram under every possible combination of temperatures, and then did a big ANOVA on the results and asked what the effect sizes were for different components of that system, and asked basically whether temperature was driving the responses we saw, the spatial arrangements and availability of patches, or some interaction. And what's interesting is if you look at the thermoregulatory ability, you see a good bit of the variation is driven by temperature, but just as much variation is is driven by the spatial variation alone, just those patch dynamics. And then some interaction of temperature adds to that. So nearly half of an animal's thermoregulatory ability is dictated by spatial features, things that aren't temperature themselves, which is really kind of interesting. You can look at how far an animal moves and you find that basically the spatial distribution of patches really drives that. But then you find that, for small animals, while temperature really drives a lot of the energetic dynamics of the animal, as animals get larger and I guess more independent from the environment, temperature itself becomes less important for driving energetics than does space. So space, again, is one of these things we've been ignoring in these large scale models. And so it's likely to lead to some erroneous conclusions when we run these things. No model is good without a test. So out in New Mexico, we created some large enclosures at the Sevieta Long-Term Research Station. Here's an aerial view. These are roughly 20 meters by 20 meters. And we could recreate all these different shade patches that I had on my computer and actually release real animals into them. So we used Yarrow's spiny lizard. It's about a 10 to 20 gram lizard as an adult. We took these eye buttons. You can take these apart, take the insides, dip them in silicone. You can actually implant these in animals and it'll measure their body temperature however long you want. So in our case, every 10 minutes, and release them into these enclosures for a few days. And we asked, could our models predict the thermoregulatory ability of these animals? And so I wrote an individual-based model that used the actual weather data during the experiments that we did. Here are the data plotted. Basically, this is a cool time of the day, warm time of the day, but also you get more variation. So we had to do a PCA axis on that. But what you'll see here is the 16 patch is this green, the 4 degree patch, or the 4 patch is in blue, this 1 patch is in red. And basically, the more narrow that range of temperatures, the better these animals are thermoregulating. Here are the observed data in real animals. You'll note it looks really similar. Our model actually did a really good job of predicting how those animals behaved. And if you look over here, this is the residual variance about like a line that would go through there. This shows that the 1 patch performed about a degree worse in its thermoregulatory ability than the 4 and 16 patches. And you may not have remembered, but when I showed you some predictions earlier, that 4 and 16 patch under these approximate shade conditions should behave similar. So our model seems to be doing really well predicting that thermoregulatory behavior. We've also done some other things, as I alluded to. We can talk about this in the happy hour. We've looked at animals interacting with each other, and we've also done things where we simulate predation. You get some really interesting dynamics with those two, but that's another time. But what I wanted to do is say, like, well, okay, we can study models, we can study these things in enclosures. Can we apply this to a real system? Now, I haven't actually gone and done a direct test, but for my dissertation work, I looked at variation in body size and how that was driven by the thermal environment. So I was able to go back to that old data and ask whether my approach could make some successful predictions, because other approaches hadn't. What's nice about this system, this is the sagebrush lizard systems, Scolarpus graciosus. They've got this really nice relationship where if you look at the length of the activity season in days, the maximum adult body size goes up pretty linearly. So it's kind of nice. So you should be able to take a model like Warren Porter's that predicts activity time and get a good idea about what an animal's body size would be. Because what there are some early papers by Adolph and Porter saying that, the longer an animal is active, the more time it has to forage. That means it should have higher growth rates and could grow to a larger body size. Now, as a grad student, I was poor. I couldn't afford to travel all across the continent to study those different season lengths. But what I could do is take advantage of a more local system. So I studied these lizards at Zion National Park in Utah, and I took advantage of an elevational gradient. So here's a low elevation site, a middle elevation site, and a high elevation site. And then the prediction that you would make from Warren Porter's model would be that animals should grow more quickly and to a larger size at this lower elevation site because it's warmer. As you go up the mountains and it gets cooler, you would expect these animals to become smaller and grow more slowly. But nature's more complex than that. I did a mark recapture study for 4 years, and what you find is that the animals at the high elevation site, while they do start off smaller, they actually grow faster so that by the end of their first growing season, they're the same body size as the animals at lower elevations. So this is sort of the opposite prediction that you would make from such a model. So first thing going through your head, it's like, what could be driving that? Okay, maybe there's local adaptation. So we do a common garden study, bring animals, gravid moms in, let them lay eggs, and we hatch these out under controlled conditions, and did a feeding experiment on the offspring to see if they grew differently depending on what population they came from. And basically, these animals are the same no matter where you grab them, which was kind of interesting as well. So what was driving the differences then in growth rates amongst the populations? Okay, maybe food. Well, every time we went out and assayed food in the environment, it was basically the same. The only differences might be that phenologically different insects emerged maybe a few weeks later at higher elevations, but nothing panned out. But then that's actually where I got some of these early ideas about spatial dynamics and thermoregulation. So now what I can do is I can take that model that I developed and ask, could it predict the correct activity times and energetics for these populations that might lead to these same patterns of growth? So fortunately, with modern day GIS, you can actually go to the web. You can collect the elevations for each of these sites that I studied. These are resolved to about 4 meters. You could get color IR images resolved to about 1 meter, and then I could recreate my study sites on my computer, and then I can apply those silicon lizards with the right behavioral characteristics and assay my environment to say, well, how long could they be active and how much energy did it take? And I can actually take into account the vegetational structure of this environment. And what's interesting, let's just concentrate here on the June, July, August. This is the low, middle, and high elevation site. What you'll notice is actually when you look at the amount of activity time an animal could achieve, not just from the temperatures, but when you're adding in those vegetational characteristics, animals at the lower elevation population or at the high elevation population have the most activity time during the season that these juveniles are going to be growing. So that's kind of cool. It suggests that the idea that Warren Porter had was right, that the longer time you have, the more you could grow. But you really need to take into account the spatial characteristics of the environment and how an animal behaves to understand it. Furthermore, if you look at the amount of energy it takes per day for the animal to achieve that, you'll notice that the high elevation lizards always had the least energetic expenditure than the ones from low elevation, meaning that if an animal actually, if these animals had the same amount of energy to allocate, the high elevation lizards would have more left over to maybe grow more. So very interesting. And it turns out, by the way, my model actually estimated correctly the amount of energy expenditure for an animal at the middle elevation population that was estimated by Justin Congdon back in the 70s. But he estimated about 400 joules per day from a double labeled water study. So the model seems to even be getting some of that right. So that's kind of cool. You know, I've never expected any model that I wrote to work. But we can now begin to ask questions about whether we could apply this to species ranges. So we can predict, you know, from spatial characteristics of an environment, some thermal map of the environment, translate that to activity, maybe get a better picture. But to do that on a continental scale seems kind of ridiculous when I'm modeling things at meter or sub-meter scales. But what I've been able to do is I can actually simulate topographies on my computer. I can create flat environments. I can give them a little more topographic relief, or I can make a lot of topographic relief. I can change the fractal dimension of that, so I can make this more smooth, these more bumpy. And then not only that, I can actually change the amounts of vegetation and the fractal dimension of those too. And so what I've done is I've designed an experiment where I sample, quote unquote, all possible landscapes. So I take a 1 hectare plot, I designate the elevational range that it will have from 0 to 20 meters. I choose a random fractal dimension of that elevation. I give it some vegetational cover and then a fractal dimension of that. So you create all these different landscapes that are sampling state space of what they could be. I'll simulate activity for a couple hundred individuals in each landscape, and I can record how many minutes are they active and how much energy do they expend. And you find some really cool results. So one, and this is the thing that's problematic for a lot of models that Mike Carney, Warren Porter, Lauren Buckley run when they're estimating activity time and trying to translate that into energetics. One is that activity at different times of the year. So this would be, say, in spring, early summer, midsummer, what you'll find is that 50 minutes of activity at different times of the year have different energetic consequences. So you can expend, over double the energy trying to thermoregulate in an environment when temperatures become limited because it's warm and there's more distance between patches. So that's kind of interesting. You have such a broad range of energetic expenditures, even though you're active the same amount of time. But also, on any given day, just by changing the spatial characteristics of the environment, you can have a broad range of potential activity times. Now, what's cool is you can look at the effect sizes, and you find that you're getting some pretty large effect sizes for things like vegetation, and still some for elevation and things. But this model can explain roughly 80 to 85% of the variation in activity and around 85% of the variation in energetics. So basically, we can shortcut the system. Instead of having to simulate a whole continent, you can come up with these relationships from linear regression that can correct from a flat surface to these more complex surfaces and get a better estimate of what these energetic expenditures will be. Now, we haven't done that yet, but we suspect that we can get a higher resolution fit for the models. And so again, it gives us some predictions across space about when and where animals could be active in a real context. Now, I'm going to switch gears here and then kind of tell you that maybe, some of what I told you is not true, and not true in the sense that these animals aren't thermoregulating and the like, but that the story is more complex and maybe it's not the only predictive thing. And so what we've been doing lately is looking at the whole life cycle. So typically here we focus just on adults, but adults are just part of the problem. So, you have an adult lizard, she may dig a nest, they have eggs. These eggs can't thermoregulate. They're stuck where mom puts them. And then, they hatch. You're a very small lizard, and you're a bit more prone to variation in temperature because you have no thermal mass. So the environment that an offspring experiences is different than a juvenile, is different than a mom. So we're trying to incorporate some of those dynamics. So here's an example of what it means to be an egg. If mom puts you in a nest and gives you a fairly shallow nest, you have a wide range of temperatures that you would experience, but the deeper you go, you get a more narrow range. So mom can really determine the thermal environment that you're going to live in, which determines developmental rates, body size, many things. Mike Angeletto's lab has done some work showing how these nest characteristics can lead to different patterns of survivorship. So he wanted to look at how acute stresses in the nest affected embryos. And what he found here was he used heart rate as an index because we think that cardiac stress is how these animals are dying, is that as you warm temperatures between 34 to 41 degrees, and this is pretty warm for an egg, heart rates start to increase. Around 41 to 44, the rates get more erratic, and by 44 to 47, basically the animals die of cardiac arrest. And regardless of where you're sampling these animals across their species range, you get approximately the same temperature for that CT, that critical thermal maximum for eggs. And what they did is they put that relationship into a model. Here's Survivorship. And they wrote a model, Ophir Levy was responsible for this, that simulated an animal's life cycle. So they estimated the amount of activity an animal could have at the surface, but they also estimated embryonic survival and growth given temperature dynamics of the soil. They incorporated this relationship into that model that they measured in the lab, and they made some predictions. And what they found is that you would see decreases in fitness up to 35% across the species range when you integrated that embryonic physiology and didn't just look at adults. And so that's pretty interesting that it's going to have that large of an effect. I'm not going to go through the individual scenarios here, but just the idea that in fact it has a big effect. Now, the thing is, lethal temperatures aren't going to be the only thing that's affecting embryos. You're going to have sublethal temperatures, so where you get warm climatic events that affect your physiology but don't necessarily kill you. And so I was interested in what effects would those sublethal temperatures have on animals. So I had a grad student who looked at this. We studied female fence lizards both in the field and in the laboratory. He went out, found gravid females. We could these females would lay their eggs. We could divide them up into individual little cups where we rear them, put them in incubators where we subjected them to different temperature cycles that mimicked nest conditions in the field. And we could look at how quickly these animals developed, what was the survivorship, what body sizes come out, how did they grow. At the same time, we did the same type of study in the field. We followed animals until they laid a nest, excavated their eggs, rearranged them into different nests. But then in half of the nests, we would actually put this ground cloth over or top of them, because that would, because this is dark and the sun hits it, it actually warmed the soil underneath by a few degrees, something you might expect from climate change. And then we can compare them to nests that had not been manipulated. All right, so now we have a lab experiment looking at how temperature affects embryos, and a field experiment trying to do the same thing. Here are the temperatures we used from data in the field from a previous year. We knew that nests in South Carolina had a thermal cycle approximately like this, ranging from about 19 degrees to 32 degrees. And we simply increased the maximum temperature a nest would experience. Turns out that's approximately how nest temperatures would change with increasing temperature. So we did a 3.5 degrees Celsius and a 7 degrees Celsius increase, because we didn't quite know what to expect. When we warmed the nests, we actually found that we could warm these things by 3 or 4 degrees Celsius. So basically it looks like the difference between a normal nest and maybe this middle temperature cycle. And then we can start looking at the effects on embryos. So here's survivorship. We found that animals at the warmest temperature in the lab had decreased survivorship compared to the cooler temperatures. If we go to the field, these were our natural nest survivorships. Now, in the nature, these things are also affected by soil moisture and other things, so the survivorships are a bit lower. But what you'll notice is, like, you don't even really have to run statistics on this. We had a much lower survivorship for these nests where we actually had the ground cover where they're warmer. And we did other things like measured soil moisture and oxygen to know that the only thing we manipulated here was temperature, and that you saw this effect of higher mortality. Furthermore, you find that warmer nests have a faster development time in the lab. We also found that in the field study. So you could see a reduction of about 20 days of developmental time just by warming the nests. But interestingly, there's also an effect on the body size phenotype. So hatchlings in cool environments hatch out larger in length versus animals at warmer environments that came out smaller. You see the same thing reflected in mass. But then what's interesting is if you looked at the subsequent growth rate over the next 30 days, you saw an effect on that too. So animals that were born to warmer nests, not only were they smaller, but they actually grew more slowly through the first month of their life. So there are going to be these carryover effects into other life stages as well. And in the field, we also noted you saw the same sort of differences in body sizes. We didn't have enough numbers to be able to do the mark recapture on hatchlings. Anyone who studied hatchling lizards in nature knows that they don't last long. All right. So what we did is we then added our sublethal effects to Ophir's model, reran it. And what we found was that sublethal warming could reduce survivorship in nests and also fitness across the range now by up to 24% than the other previous model iteration. So here are our new predictions. What you're going to see here on this last panel, this is the difference between including a model that doesn't take into account sublethal effects versus the one that we did. And what you find is by not doing that, you're really underestimating how well populations do in the southern part of the range where it's going to warm up more quickly. So kind of a cool result. So again, this is having a big effect. Now, another thing that we're interested in is how whether an animal can actually track climate well enough, quickly enough to adapt. So what we did is we looked at 3 populations across the species range, and we did a common garden study where we simulated each environment for these animals and did a lab reciprocal transplant. So what we do know about these areas is that nest characteristics vary across the range. Here in South Carolina, you actually have a rather shallow thermal cycle, and this is because the environment's warm enough that the moms have to lay their nests underneath the forest canopy. They are choosing slightly the warmer temperatures underneath the canopy, but if she just picked a nesting spot by random, it wouldn't be too different. So basically what's probably going on is they're picking relatively cool parts of the environment, and those are still pretty warm for them. If they went out into the sun, the nest would overheat. Now, if you go up to New Jersey, it turns out these animals actually experience much warmer nest temperatures. They have a much higher variation across the day. And this is because basically here's what a random nest would look like if you just went out and poked random holes in the ground at nest depth. You basically get cool temperatures, and if females chose areas out in the forest, their eggs would never develop. So they go out and they actually choose the hottest spots in the environment, the most exposed, and you get this type of cycle. What's interesting is that means in the future, as climate's warm, they'll probably move their nests to the forest, and their nest temperatures will actually get cooler in response to warming temperatures, which is kind of cool. But anyway, we know what the thermal cycles for nests in New Jersey, for North Carolina, and for South Carolina look like. We got moms with eggs from each of those different sites, and we reared the eggs from each site across nest temperatures from all sites. So we have all combinations. So South Carolina lizards reared in South Carolina nests, North Carolina nests, New Jersey nests, right? And then we did the same thing that we looked at in the previous experiment. So we looked at survivorship, and good news, regardless of where you're from, survivorship is high across this different cycle. So what we're basically doing is a space by time replacement. So we sort of suspect that, New Jersey nests, in some amount of time will look more similar to North Carolina nests and eventually South Carolina nests. But what you find is, regardless of where you're from, you survive equally well in any nest type. If you look at developmental time, this is South Carolina, North Carolina, New Jersey nest conditions. Again, coolest nest condition, warmer in terms of that maximum temperature. You find that animals, the thermal sensitivity of developmental rates is pretty similar across populations. No differences, but if you warm the nest, they develop more quickly. Now, this is looking at body size. In this system, it's well known that these animals follow a temperature size rule. So in northern latitudes, animals are typically, they mature later to a larger size, have larger offspring, and you see that reflected here. So in the warmer environment, they're smaller. Middle latitudes, they're a little bit larger. At the most northern extreme, they're the largest animals. But within a site, South Carolina animals will still grow to be the largest at cool environments, the smallest in warmer environments, and you see that across the range. Same things reflected in hatchling mass, and in growth rates, you also see that the same thing. So slower growth rates in New Jersey overall, faster in South Carolina, but they grow the most slowly in those warmer, when they were born from warmer nests. So you're still seeing those carryover effects. So, what's that going to mean for climate change in the future for these animals? Well, here is our New Jersey nests. What's going to happen with climate change is moms, if they have the ability, when the environment warms, they're going to either choose deeper nests or they're going to move into the forest. And so actually the effect that would have on nest temperatures is they're actually going to decrease, even though there's a warming environment, because they have the behavioral capacity to change micro habitat sites where nests occur. Now in the South, it's a whole different scenario. In the South, this is what the nest temperature cycles look now, because females can't go any deeper to the forest than they are, and we don't think that they actually will modify nest depth, though we could talk about that. We have some interesting patterns of where animals put nests. But regardless, we expect just their whole thermal cycle to rise. Now, what's that going to mean? That means here in New Jersey, they're going to have cooler nests. Their babies could potentially grow out to be hatch at a larger size, grow at a faster rate, and the adults are going to be doing fine because this is the northern part of the range. It's cooler already. New Jersey could be a place in the range where you see accelerated gains. Now here in the South, because nests are going to get warmer, there's going to be higher mortality. These animals are going to hatch out at a smaller size with slower growth rates and maybe not be able to reproduce their first year. You could see accelerated losses because of the demographic effects on change. And so these are things we're starting to look at. In the future, we're adding a genomic component to this. So we're going to use some gene expression studies. We're going to repeat these experiments, but also add water stress and look for the potential for different populations to respond to these stresses and whether there's any adaptive capacity for that. And that's something we could talk about later as well. But what I hope I showed you today is, we're really trying to expand our capabilities in terms of modeling and understanding what types of data we need in these models to make better predictions across the range. So hopefully if we went back to our fence lizard models compared to what we did in NCs, a decade or so ago, these models that we have now are doing a much better job at predicting the actual range. And so I just want to take this time to wrap up. Thank funding agencies, NSF in particular, along with the NCs and NESCENT working groups we had, and then some particular people. My long-term collaborator, Mike Angeletta, his former postdoc, Ophir Levy, who I collaborate with a bit now on some global climate data sets. And then my past students, Eric Riddell and Mike Carlo. Mike did all the juvenile or all the egg studies for the last part of this talk. And Eric is now a professor at Iowa State. He could give you a whole really cool seminar on the work we've done with water loss and salamanders, what that means for range dynamics, and the genomic capacity for plasticity and things like that. So he's been doing some really cool work and has had some great papers recently. So with that, I'll wrap it up. I think that kept me at time too.
Thanks, that was great. I'm just gonna, so I have questions.
Okay.
But oh yeah, first, that was wonderful. We should all clap. I don't think we have the clap emoji though in this particular meeting style, unfortunately. So I'll just clap for everybody. There isn't any questions in the question answer. So why don't I go ahead and ask a question while people in the group, if they have questions, should please put their questions into the Q&A, and then either Brooke or Changmeng or I can read those. Actually, there's one question. So I'll let that go first. Actually, there's two questions. So the first question is from Doug Woodhams, and he says, cool talk. Can you comment on the physiological differences you might expect by raising lizards in fluctuating temperatures versus an average constant temperature?
Yeah, instead of talking specifically about lizards, I collaborated on a study looking at striped marsh frogs from Australia. And we did a study where we measured the thermal reaction norms for growth rate and developmental rate and tail regeneration, or I guess tail through development where that goes away. And we estimated, a thermal reaction norm. And then we actually had other tadpoles that we raised in different fluctuating environments. And so we've actually measured their growth rates in a thermal cycle, one that was relatively cool, one that was a little warm. And then we asked, could we actually predict the growth rates or developmental rates from the temperatures that were predicted, at constant temperature from those reaction norms? And it turns out, no, you could be off by 50%, as much as 50%. And usually you over predicted, but it wasn't always clear that that was going to be the case. So we have a paper in Journal of Experimental Biology. You could look at that. But basically, it says that we really probably need to be measuring these things over some temperature cycles, and then it's going to be a little harder computationally to back out what those thermal reaction norms that you should be using are. So you can basically, if you have some statistical distribution that you have shape parameters, you could actually say what are the values of those shape parameters that give us the best fit to the data that we collect under variable temperatures. And to some degree that approach works, but still not always. The boogeyman in all of this is that these animals, while they have these acute responses to temperature, are also having more chronic responses. So across the season, you have that sort of right shift in the thermal reaction norm, and then when it gets cool, it goes back left, and that occurs, with some time lag. So it gets hard, but the one thing that we seem to know, and there's an emerging literature on this now, that basically it's really hard to predict the response of animals under variable environments from constant temperature data. So that's one of the things I try to encourage people to go out and measure. It's not sexy, but it's necessary.
So thanks. Yeah. So I'm still here, but my video is kind of messing up my Zoom, so I just turned it off. So I'm going to ask the next question as a disembodied voice. So Ron Eder asks, Thanks, Michael. Very cool. Any sense of why the common garden experiment indicated lizards showed no adaptation and growth at different elevations in Utah? For instance, too much gene flow among elevations, etc.
Yeah, I mean, I would imagine that that's probably it, because, you know, while the populations I studied as a crows fly were, you know, certainly divided, you know, there wasn't gene flow from them. If you're going up a mountain, though, these lizards are pretty widespread. There's probably enough contact, you know, that you're always getting some low level gene flow across populations. So I think that's probably it for these. When you compare it to something like the eastern fence lizard system, the reason you see like the opposite pattern, where you see local adaptation, probably is because you also have predators that are like mortality rates that are also correlated with latitude. So in the south, you just have tons more bird and reptile predators on lizards. So you have, you know, higher mortality rates. You're going to select for faster growth and the like, versus in the north, you lose a lot of those larger body ectothermic predators. And so it pays off for females to delay reproduction and the like, and you get that rule. Whereas in the west, that doesn't occur as in those systems occur as much. So yeah, so you might expect, just because of the gene flow that you see between those populations, that's why there didn't seem to be local adaptation. But I was as surprised as anybody when I was getting the data as a grad student that they were coming back exactly the same, because the growth rates you see in the field are very stunning. When you see a small animal at high elevation that's maybe a gram and a half, and the same age animal at low elevation is 3 grams, that's pretty drastic. But then by the end of the summer, they look the same. You know, that boggled my mind. So really just sitting out in the field and thinking about, you know, what these animals are doing. And just one day it hit me, it's like, oh yeah, when I'm at my low elevation site, these animals are stuck under the bushes. They can't get out. But if you go up to the high elevation, you kick a bush, you know, it was Frogger. They were everywhere. And so it's like, maybe it's just like these realized differences in activity because of the structure of bushes. Maybe that's what's driving those differences. And I think I looked into it.
So the next question, I think we still have a little bit of time, right, Brooke? So the next question is from Rob Stevenson. He says, fun talk, thanks. In the simulations, how were patch size, movement rates, and body size scaled?
So in those, let me see if I can, there's an easy way to go back and up. Maybe not. Each of those pixels in that simulation was a half a meter, I believe. The way that worked is I gave animals a body size so that there was some relationship. There's a paper by Grigg et al. In 1975 or something that estimated the heating and cooling constants of lizards of different sizes. So I used equations from that to get the heating and cooling rates. Basically what I did for that model is at every time step, you assessed what your body temperature was. If it was inside your preferred range, there was some probability that you remained still, but you could still move. If you're outside that range, there's a probability that you moved, which was higher. What you did then is you sampled points around you. I gave the animals a perceptual range, and basically your perceptual range to detect the temperature decayed as distance goes away from you. So sort of like a beta distribution where it's flat further away. And then I used a circular von Mises distribution to determine whether you could sample equally well around you, or if you had a more forward looking approach. And so I've done these simulations for all different types of things where you're sort of more of a sit and wait and you can shuttle, or if you're like a Spedicellus and you're just moving about the environment all the time. You get the same type of results, just the magnitudes change a bit. But the only thing that changes, like when you're scaling up to a larger animal's body size, is that heating and cooling constants change, so you get a lag. And with these grids that I created, especially like even the ones with the 3D topography, because they're made from the fractal dimensions, you can actually tessellate those infinitely. So they just repeat, over themselves. So I just allow the animals, if they're larger, okay, yeah, the maximum movement distance was longer, so they could just move further distances, but the habitat structure itself stayed intact. I think hopefully that answered what you were getting at.
Well, I'll have to, since I'm only the proxy. So can we ask 2 more questions or no? Okay. So so Rick Kessley says, in New Jersey, it sounds like you said the critters will have deeper nests. So are the adults responding to the needs of the offspring as they develop? Not sure if I'm clear. If not, I will try in the happy hour.
Could you repeat the first part of that? That the females do what?
In New Jersey, it sounds like you said the critters will have deeper nests.
It turns out they have about the same depth nests across the range. What they seem to do from the nests that we've excavated is, this is a lizard. It basically digs a body length deep, so her head's poking out. And maybe they could dig deeper. We don't know that they can. Maybe they can move the nest to other places. We don't know. That's what we actually model those things to say like what would happen. What we do know, I don't know how much time we have here to answer this, but we know that females like in New Jersey they'll all go to the same site to nest. Like they go to these forest breaks, right, where these old railroads are. So they don't nest out in the forest where the home ranges are. They'll move up to half a kilometer to get these hot spots. What we know in South Carolina is that they nest kind of randomly in the forest, but we know that they're going to almost exactly the same spots. So like when we're excavating a nest, we'll see shells from the previous year. So probably, mom's going back to the same place or some file patchery. But we know that later in the year, like for the second clutch, she moves to a different spot. So maybe she's switching positions because maybe it's warmer now. You need to nest in maybe a slightly cooler spot. Maybe she's able to do that. We have limited data, but it looks like that might be what's happening. So we have lots of ideas about things we would do to females in terms of trying to confuse them by moving them to different home ranges, by taking eggs in places where they might put them and shuffling them around to try to disentangle what's going on with that. But it doesn't appear that females are really modifying nest depth. And given some things we know about, when they're, in the South here, they'll nest during the day, during night, whatever. In the North, because of where they're nesting, they have to nest at night because the spots that they choose, they would overheat during the day. So it's kind of weird watching a lizard excavate a nest at 2 a.m, but that's what they do. They may have lost the behavioral capacity to do anything else in the North, because selection, as they move back north in the ranges, from the last glacial retreat, we don't know whether that behavior is hard set or whether they still have the capacity to make more choices. So who knows?
Yeah. So should I ask this one last question, Brooke? I think so. So the last question is from Breck McCollum. Do you expect the southern lizards who experience sublethal temperatures more frequently will be better adapted to withstand extreme temperature events than the northern lizards?
Yeah, it's hard to know. So the northern nests experience broader ranges of temperatures and higher temperatures than the southern ones, right? So maybe they're better adapted for those things. But in the South, there's going to be more genetic variation. Just because that's sort of the core of the range. And so maybe there's more adaptive potential. I think because those animals are experiencing sort of, more shallow, lower amplitude thermal cycles, they may not do so well with these temperature spikes as ones in the North do. So I actually think that in the South, they're more, even though they on average experience warmer temperatures, you would think that they actually may be less tolerant to those spikes. And so sublethal effects, I think, are going to have a really big effect in the southern populations. Where in the North, it's, they're in some sense, they may be buffered to it. So, we wonder how, to what extent some of these thermal characteristics on growth are sort of locally adapted up the range. So, that's why, in the future, this summer, we're going to go and take tissue samples from bunches of populations up the East Coast, look for variation across the genome, see if we can look for places in the genome that have been that showed evidence of natural selection. Are those things thermally sensitive regions for developmental traits and things like that? So, and then we're going to do gene expression studies on those sorts of things. And hopefully we'll be able to pull out some of those answers to know which populations might have the best capacity to deal with, what future temperatures would look like. So, it's up for debate.
All right, thank you. That was a great question and answer session. It was so great that I didn't get to ask any questions. So I guess that's good. I put a link to the social hour into the chat that everyone should be able to see, like all panelists and attendees should be able to see. So if you want to go to the happy hour, and I hope that many of you will, you can just go and copy the link out of the chat.
Can you do that? All right.
I also emailed it to you, Mike.
Okay, cool. That's what's out
now, just in case.
Okay.
Yeah, Mike, feel free to take a short break before you join us in the happy hour if you feel like you